I said again, most lipids are CHO, but really they're mainly CH. I said that, right? CHO, but mostly CH, carbon and hydrogen, long chains. How about if I just write it on the board like I just did? All right, so CHO, mostly CH, okay? Mostly carbon and hydrogen chain. Now you should already get this. Now, as a quick review from the previous slide, you have this molecule called glycerol, which is how many carbons? How many carbons? It's literally drawn for you in front of you in your study guide. 3 carbons. It's 3 carbons. It's really easy. It has an OH group coming off each carbon. Not hard. Some hydrogens here. It's easy. How many carbons do you see on this behemoth over here? How many carbons do you count? How many carbons? 3. What molecule do you think all this was attached to? Glycerol. Thank you. Glycerol is a foundational molecule that we build lipids, a lot of lipids on. Triglycerides are 3 fatty acids on a glycerol molecule. Is kind of the base of a lot of this lipid chemistry that happens in your body. So 3 carbons. This is a glycerol molecule. This is not a fat, right? It's not a lipid. It will become a lipid, though. I'm going to say this. I said this before in the previous slide. I'll say it now. Lipids are not polymers. They are not polymers. They don't have monomers. They're the only macromolecule in your body that is not a polymer. Proteins are polymers. Nucleic acids are polymers. When you store carbohydrates, carbohydrates are stored as polymers. Are we clear? Lipids are not. Lipids are not polymers. So it's just a long chain of carbon and hydrogen. So what is this? Carbon and hydrogen. The white ones are hydrogen, the black ones are carbon. What are these? Carbon and hydrogen. These are some oxygens, and there's a phosphate at the end. This is this tip here. This is what sticks out, you see. This is a drawing of what you see here. You see all these oxygens? Those are these oxygens here. These 1, 2, 3, 4. 1, 2, 1, 2, 3, 4. This is actually phosphate, 1, 2, 3, 4. And this is phosphate here with phosphorus in the middle. So phosphorus would be in the middle here. Let me color it in. I shouldn't do it. I guess I should do some other color. So this would be phosphorus, right? You can't see it because it's a space filling model. And then it has oxygens attached to it. 1 oxygen here, 1 oxygen attached to it here, 1 oxygen attached to it here, sharing electrons, right? And 1 on the other side that you can't see. Is that clear? So they got 4 oxygens around 1 phosphate. Well, rather than trying to color it all in, right, what we can do is we can draw it in as a structural formula instead of space filling model. And we can see that there's nitrogen in this phospholipid has nitrogen, right? This phospholipid has phosphate. This phospholipid has a glycerol molecule as a base. To the third one, if you added a third fatty acid, this would be a triglyceride. Do you see that? But instead of adding another fatty acid, what did we add? This group here, you see it? We added phosphate plus this little section, this little tail here. The key is phosphate. That's where we get phospho. What? Phospho. Go ahead. Phospholipid. 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 It's phospholipid. This is an incredibly important molecule. Oh my God, it's so important. So you can guarantee you it's going to be on the test in a bunch of different ways because it's really important. Every cell of your body is made of phospholipids. Every single cell of 1 trillion cells that make up who you are, all of them have phospholipids in it. Incredibly important. And their properties are so cool. So we're going to do an activity, we're going to do a lab next week that's going to have you build a lipid out of some soap and a string. It's going to be very cool. I like it. And it's going to emulate, I think everything's cool. You guys are just lame, so I don't know. I'm kidding. What's interesting about phospholipids, what do we know about carbon-hydrogen chains? What do you know about them? They're what? Give me something you know about these carbon-hydrogen what do you know about lipids, carbon-hydrogen chains? I say the word lipid, in your mind, you should be thinking carbon-hydrogen chains. Lipid, carbon-hydrogen chain. Maybe ring, but mostly carbon-hydrogen. Mostly carbon. That should just be, you should say it in your nightmares, in your dreams. Right? Carbon-hydrogen chains. Carbon-hydrogen chains. Should be just linked in your mind. So when you think of a lipid, you think of a carbon-hydrogen chain, what are you thinking about? What kind of properties does it have? What are you thinking? You don't think of anything? Okay, that's good. Yeah. Doesn't dissolve in water. Excellent. Perfect answer. Exactly what I was looking for. So what is it? By the way, excellent. You notice you're the only one that did it? That said something? You should be proud of yourself. Doesn't dissolve in water. What does that mean? It's what? Nonpolar. Nonpolar. Doesn't dissolve in water and is nonpolar. So another word for nonpolar. Nonpolar. Does not dissolve in water. Can I use the word does not? In quotation marks. Like, I'm writing it down, so I think you should be. Water. Can I write that? Does that make sense to you? Yes, ma'am. How can I help you? So, doesn't like water. Doesn't like water.
No, we do not.
I can't believe I did that. Tell her.
He just said lipid or not.
I swear I didn't.
Your list. Are you okay?
You talking to me?
Yeah.
I mean, am I okay?
And what is called a gobbler?
Huh?
A gobbler. I don't know how to say that word.
A gobbler?
It's GLOB.
Oh, globular protein?
Yeah.
Globular protein. No, the globular proteins are proteins that are, this is not a protein. It's a lipid. Right? Lipids are, but it's not a full lipid, right? It's a what? Phospholipid. But the lipid part, what did we say about lipids? They what? They don't dissolve in water. So another way of saying that is they don't what? Like water. Well, it turns out there's this Greek word. I don't know if it's Greek or Latin, but I'm pretty sure it's Greek. There's this word that's, and you'll have to ask this the Greek Latin teacher. Philic. All right. There's this word philic. Philic means kind of a brotherhood or love, right? Then there's another word that's phobic. Phobic means dislike or fear of, right? Is that okay?
Like phobia.
Like a phobia, right? Like I have a phobia against, or right? Water. With water, what do we know of is a prefix that starts that water starts with, and most of the time we use with water. Hydro. So we can say hydrophilic or hydrophobic. So either they're afraid of water or they love water, right? So philic means love. And either they love water, or some of you are staring off into space, I hope you get it. So they love water or they hate water. They love water or they hate water. So now, what is going on? So how do we apply this to what I'm saying with lipids?
Period.
So what does that mean?
What you mean? They just said it.
I'm saying we look at this molecule. We're looking at this molecule. Right? And we have this long molecule and it says, how do we apply hydrophilic and hydrophobic to it? What are we going to do with these long carbon hydrogen chains? Which is it going to be? So you're saying these long carbon hydrogen chains, these lipids are hydrophilic. They love water. Are you calling phobic?
Yes.
And we all agree now that it's phobic?
Yes.
Okay, so it is. They just literally said philic, man. Are you not listening to your fellow students? Listen, don't contradict.
When they were saying phobic, they said phobic when you was on your computer, and then when you just turned it around, they said phobic again.
They said philic a minute ago.
Who's they? Rabina?
Oh, let me pause this and rewind. So I was right, and you did say philic, and then change it to phobic.
It's okay, everybody.
So anyways,
I got y'all.
the bottom line is, you'll listen to it on the YouTube. I'm sure you're probably wrong, but it could be that I'm wrong, possibly. So anyways, when you listen on YouTube, you guys can figure it out, see what you said or not. But anyways, these are hydrophobic. They're definitely hydrophobic. They don't like water. They're afraid of water. So if you put these in water, they're going to try everything they can to get away from them. Could we close the door, please? This side over here is a phosphate head, phosphate, that's phosphorus with 4 oxygens. What do you notice about that phosphate head? Look at that gray area. What do you notice about it?
It's big. It got a lot of oxygen.
Okay, that's a lot of oxygen. What do you know? Look carefully. We're talking about water, we're talking about polarity, we're talking about dissolving or not dissolving, liking or not liking. Think about the context of the question.
It's a lot of
Now look at that phosphate and look at it and tell me, what is it you notice is going to impact all of this?
the hydrogen bond that's right there, like.
Well, you don't see any hydrogen bonds here. What do you notice? Is that door closed? Dang, these kids
They
are
having
loud,
fun,
man.
boy. Let me go up there.
What is this now? What do you notice?
It's oxygen and hydrogen. Everything.
Oxygen, hydrogen, everywhere's good. You don't notice anything else, seriously?
I promise to God I don't.
Now I'm going to go on. I'm going to say the answer, and then I know certain people are going to say, "Oh, they said it. They said that. They said that." I know you're going to say it. I know you are. I know you're going to
What
say
is
it.
it? Come on, for real. What is it?
There's a negative charge here. There's a
Oh,
charge
I
over
didn't see
here.
that.
Oh, you see? I knew you could say.
I
swear you
said
guys.
I seen it.
You
I
guys
didn't
try to
say I
switch
said
up this
it.
story all the time. So anyways, you got charges, just like sodium chloride, right? Just like things that are polar, things that are charged, what do we know? Are they going to be hydrophobic or are they going to be hydrophilic?
Oh, I did not see that.
Now somebody said phobic. I heard that. Am I lying? All right. Something's charged, something's that's partially charged or fully charged, like ionic. Like in this case, this is ionic, right? So now we got, what do we call that? Do we call that hydrophobic or hydrophilic? Is this going to love water or is it going to hate water? Is this hydrophobic or hydrophilic? Why do you say hydrophilic? What, you're
Where?
cheating. But why is this hydrophilic? Why does this love water? The charges, that's right, because it's charged. It's positive, it has a negative. And what does water do? It dissolves things that are polar or charged, right? That's why it dissolves water and sugar and polar dyes like what we did in chromatography. So when we're talking about this, we're talking about this as being hydrophilic, as a couple of you noticed it's in the name, because it's there on the side, because some people actually read English.
I can't
So it's in front of you on a piece of paper that I handed you. Wait, oh, you don't have it? Oh, you love.
It's in there.
It's in your book. That's a good place for it.
It really
So,
is.
anyways, this charged head is hydrophilic. Hydrophilic. It loves water. So this one's loving water. This part's hating water. So these molecules have two sides to them. They're so big that these are no longer polar or nonpolar. This is partly polar, partly nonpolar. Right? Partly, and we don't use polar here because this is actually charged, right? So we use ionic, right? But in any case, this is water loving and this is water hating.
So what's happening?
Yes, question in the back?
You said that water dissolves things that have charges. That's what you said.
Yep. Do you remember salt? Do you remember us doing that? You know, everything I do with a reason. I don't waste your time. I don't waste my time. So when I had you do that experiment at home with the balloon, and I had you take that salt and pull it up, that's so you can recognize and remember that salt is ionic. It's charged. And we talked about water being polar, which means it doesn't have a full charge, but hopefully by now, everyone in the room, today, as of today, everyone in the room knows that if it's charged or polar, water will dissolve it. And if it's non-polar, it doesn't have any charge, water won't dissolve it. And hopefully by now, everyone in the room knows that long chains of carbon are non-polar and or hydrophobic. And hopefully you get that. All right, so let's move on. That would hopefully not be an issue moving forward. My guess is it will be until people get some questions wrong on a quiz or a test or an activity, and then they start to process it. My hope is that you are a wise person, not just a smart person. And remember what I said about smart, or maybe I didn't say it to this class, I don't remember, but smart people are people that learn from their own mistakes. Wise people are people that learn from other people's mistakes. So I'm telling you right now, a common mistake that happens is people mix hydrophobic and hydrophilic up all the time. They don't study, they don't review, they don't recite, they don't reflect. If you're not doing these things, you're not going to learn this stuff. Nothing that's worth learning is going to come as easy as you would like. So now you got these molecules. They have long chains. There's usually two of them, and then there's this head. Now I'm drawing a head on it, and the head is going to be what? Hydrophilic. And then I'm drawing, that's the head. And then you have the tails. There's two of them. Do you notice there's two of them? Is this one saturated or unsaturated?
Saturated. Wait.
Is this one saturated or unsaturated?
Unsaturated.
Unsaturated, because there's a double bond. This one's saturated because it's a straight bond. But, so really I should be drawing it, to draw it correctly, I should take this and put a kink in it, right? Like that. So that's a hydrophilic head, and you have tails that are what? Hydrophobic.
Oh, I see a
And you know what I was about to write? Probic. Could you shh? So you put these molecules together in a glass of water, and you're going to do this. In a sense, anyways, you're going to do this. You put these, So you put these in water, what do you think's gonna happen?
Put with the tail.
Put this, a bunch of these molecules, take fat, oils, whatever, a phospholipid. You take a phospholipid. If you put oil, if you just put lipids, triglycerides or lipids, right? Just what you would get, because they're less dense than water, is you get this little layer of oil on the surface. You know that. You see it when you see the rainbow thing on an oil slick. It just lays on top. It doesn't sink to the bottom. Right? But because they don't mix, because the water won't dissolve the nonpolar lipids. But here's this molecule, now it has a phobic head, a philic head, and a phobic tail. What do you think is going to happen?
The tail gonna be on top.
I love it. The heads will stick to the water, will hydrogen bond with the water, correct? And remember, water's out here. Obviously, this is not to scale, right? Because there would be billions of these things, except I'm not drawing out a billion of these things, all right? So you need to put things in perspective and understand when I draw these. All right, whatever. When I draw these, you have to think three-dimensionally. I'm only drawing it two-dimensionally. I'm only drawing one slice of it. This is all the way around, okay? This is a round. So it'll be a ball, right? It'll be a sphere. I'm only drawing a slice. I'm not drawing billions of water molecules because I'm not going to do that. But there would be billions out here surrounding it. We call it hydrating, right? Or dissolving. Do you agree?
Yeah.
This is what dissolving is. We went through it. There's a reason we went through it. We're getting there. We're finally getting to the biology of all this.
Yes.
So your phosphate, remember this head is a phosphate. It's that part of the drawing that you see here, right? It's going to point towards the water. What's going to point inwards? What's going to point away from the water? The tails, that's right.
I'm dead.
And these tails, they love each other. Why do they love each other?
I don't know.
You do know. That's the sad part. Yeah, you do know.
How you gonna know?
So they don't like water, so they're not what? They're not polar. They're and how do I know that you know? Because what does nonpolar do to nonpolar? They dissolve each other. Like dissolves like. All right. So this is what we call a micelle. A micelle is what forms when this occurs. Essentially, you have a circle of hydrophilic heads pointing outwards and hydrophobic center pointing inwards. So micelles are just kind of a outside. Can we close that door? The outside of the micelle is made of hydrophilic heads. The inside is made of hydrophobic tails. So what you really end up with, guys, is this what you end up with is a ball of fat. If you looked at it from the outside, let me redraw this in blue because just to keep the color coding the same. If you looked at it from the outside, you just see this kind of ball. It's a sphere, right? Oh, man, this keeps growing. All right, so you'd see a sphere, and the heads would be pointing towards you. The tails are pointing inside. If you cut it, you'd use a knife and you cut it, what you would see is the tails would all point towards each other, and the heads would all point toward on the outside. Of course, on the other side would be all blue, right? With the tails coming off, and all the tails are all going to dissolve each other. Two tails for each one. So this thing is dissolved in water. If you just had just the fat, it wouldn't be dissolved in water. Now I'm going to give you an example of something like this happening. So let's say for a second, you're eating a hot dog, which you have a brand new white shirt. It's your white, blue CSSM with a white, like, hornets and all that stuff you guys love. You know, the big hornet, big yellow and green hornet on a white background. You're like a brand new jacket. You know, it's cool, it's white, because you like white, I don't know. And we're all out at Cedar Point. Everybody's having fun. And you're eating your hot dog or some greasy chips, maybe some mustard or something crazy, right? Something with a lot of fat. And you drop that fat on that big chunk of fat, whatever it is, could be nacho cheese or it could be oil from the French fries, on that beautiful white jacket, sweater, whatever it is. So here you got your fibrous proteins, right? That's your jacket. Is that correct? That's what it looks like, something like that? You're looking at your jacket close up?
Yes. Yeah.
It looks something like that. And in the center of it, you went and dropped some fat. What happens to your jacket? It's going to stain it. You got to get that out. You can't use water. I mean, water might help a little, but it's still going to be. It's just going to be there. Water won't dissolve it because it's fat. Well, you could spray it, or you could use something called detergent. And what detergent is polar heads with nonpolar tails using phosphate. That's why detergents have phosphate. And what happens is that detergent gets sent in there with the water. What is it going to do? What are these lipid tails going to do? It's going to dissolve in the fat. The fat's going to dissolve in the tails. So you mix it up enough with the wash cycle of your washing machine, and what happens is all the fat gets put in a big, gets surrounded by gets dissolved by the by your detergent. That's what your detergent is. And away it goes. And when your spin cycle goes, out goes the water with all the fat globules, with all the fat, with all the detergent, all that stuff goes out. That's what bubbles are. Bubbles are nothing but the fat from the detergent. By the way, soap is made from fat. I don't know if you knew that, but you just take lye, which is a charged head, and you add it to a lipid tail, fat. And it makes this partly this two-sided molecule, a hydrophilic and a hydrophobic end. And if you have a hydrophilic and hydrophobic end, you can dissolve fat and allow fat to be dissolved in water. Yeah.
Why does the soap bubble?
Well, what happens is, well, that's the next. Let me start next, because actually it's a good question. But there's something else that happens. That's if you just had my cells. But that's not what happens. Your cells are not balls of fat. That's not what your cells are. So it turns out if you add a bunch of these together in the right circumstances, what you get is a bunch of heads. And of course the heads are all pointing towards the water, right? I'm going to leave spaces in between, because otherwise you just have. You have a bunch of heads. And then you have these tails. All right? The tail, now I'm not gonna draw it all the way around, but you would draw it all the way around, literally, right, in a circle. That is a close-up of this. This is what? What am I drawing here?
A chain.
A cell. This is cell. This is your cell. These are all, the trillion cells are all pretty much look something like this. It might have some weird shape or some shape like this or something like this. Yeah, like your epithelial cells look something like this. Squarish, right? Ladies, now I'm looking at you. Now I'm looking at you. Just so you know. So here's your hydrophobic tails, hydrophilic heads. See, now you got what's called a bilayer, where this was just a ball with a center that was hydrophobic, outside was hydrophilic. What do you got here? You got a center that's what? Hydrophobic. And you got, I hope you're talking and then you're going to say you don't understand. It's really going to be irritating. So hydrophobic inside, and outside you have what? Hydrophilic outside. But also, what do you have on the inside? What is it on the inside? What's on the inside of a cell? Water. So it's water on the inside too. So the center doesn't allow you to dissolve water. But there's water over here, there's water over here. So what do we got here? What does this act? What is this? This is what we call a membrane. This is called the cell membrane. Specifically, it's another name for it is plasma membrane. So this is it. This is where we're starting the very first, most basic unit. You're sitting there reading your planner and not writing notes. I'm making a note that you're not writing. You're reading your planner instead of writing notes. So this is cell membrane or plasma membrane. So now the question that was brought up is how come bubbles form? Well, what happens is that you form a membrane. And inside bubbles, there's something called air. And you can form membranes as if one of you guys earlier said, well, you could have all the heads like this, and then all the tails could point upwards, right? That could happen. That's possible. But with cells, with living cells, you have what's called a bilayer. We call it a bilayer. Do you see why we call it a bilayer? Why do we call this a bilayer? No. Bi means what? What does bi mean? Two. It means two what? Layers of what? Two layers of what? Lipids. Membranes. A lipid, it's called actually a lipid bilayer. So that's three words for the same thing. Cell membrane, plasma membrane, lipid bilayer. Two layers of lipids. You can have one layer of lipid and fill it with air. You get a bubble. That's what you're going to be working with tomorrow. Not tomorrow, I'm sorry. Next week. All right? You're going to actually make these. I'm not going to get into what, but I know what the heck I'll tell you. I'll give you some foreshadowing. You can take and you're going to make a string. You're going to tie a string. You're going to wet it. You're going to have a circle. You're going to have a circle. You're going to float it on a bubble. So there's going to be fat. You're going to have, out here you're going to have, a bubble has phospholipids, right? So you have your phospholipids out here. You're going to have phospholipids inside. You're going to have the string floating in the center. then what you're gonna do is you're gonna get rid of the center. You're gonna pop the bubble there, but the rest of the bubble's gonna be there. So what does that create here? A void, or what's another word for void? A space? Well, it's a space in the membrane, which acts as what? What's it gonna allow you to do? Get things in and out. So this is your door. This is a door or window, right? That's how things get in and out of your cells. How do you get something that's dissolved in water? So if it's dissolved in water, it's what? Polar or charged. How do you get that through a membrane that's nonpolar? You need a hole. You need a space. You need a door or a window. So we're going to talk. So you're going to create one in a bubble, because I can't build the cell for you and create one. So we'll just do that. It'll be quick, hopefully. Though sometimes you kids surprise me. So that's called a lipid bilayer, and of course you have to have a way in and out of a lipid bilayer. So let's hope that's clear to you. And now you understand what a bubble is. Why does soap make bubbles? Because it's the membrane, the fats all dissolve each other, and they fill up with air, so they make a bubble. That's all.
Why does soap clean you though?
What's what?
Why does soap clean you?
We just did it. So you have fat on your clothes or on your skin, right? That's what makes it's fat. So the water, anything that's polar or ionic, right? If I'm at the ocean, I don't know if you've ever been to the ocean, but if I'm swimming in the ocean, I get out of the ocean, I'm being full of sand and salt. These things are sand especially. But salt too. Even if I don't have sand on me, I have salt all over me. You ever swam in the ocean before? It gets salty, especially if it's hot, a hot day, you'll get layers with salt. I mean, I used to swim in Puerto Rico all the time, and they'd come out and they'd be like, ah. But you go in the shower, I wouldn't have to take a bath, I wouldn't have to use soap. So I get in the shower, the water touches my body, what happens to soap? It dissolves. It's ionic, the water's polar, it dissolves, it goes away, I wash up, I go get my hot dog or I go get my coconut from the guy in the hut on the beach and sit underneath the tree and drink my coconut in peace, right? That's what we do. We bacalaito and coco. It's a fish. It's a codfish, delicious. It's fried. They got a big kettle. They fry it up and good stuff. But anyways, the bottom line is that's the life. That's why we don't need to use soap to wash off salt. If you got sugar on your hands, just use water. The sugar will dissolve right away. But if you get dirty, if you get all kinds of nonpolar stuff on you, then the water hits your body. It'll stick to your body, but it won't dissolve the stuff. You're dirty. That's why it feels dirty. You feel greasy. You ever feel greasy or get stuff like or waxes on you? You try to get that stuff off of you. You need to use soap, because soap has this bilayer. Soap has, well, it might not have a bilayer, but it has this hydrophilic and hydrophobic part that'll dissolve the fat. Does that make sense?
Yeah, like Vaseline.
Like Vaseline. If you want to get Vaseline off of you, you can't just stick your hands in water. It won't work. You need soap to help you get it off. You ever working with a car? Anybody here ever working on a car, kind of work on something? You get grease on you? You can stick your hand in water all day long. There's still going to be some on you. You'll touch stuff, and it'll mark up everything. So what you do is, what do? You use soap. And what's soap? It has that hydrophilic side and then the hydrophobic side, so the grease gets trapped in the hydrophobic pocket, and the hydrophilic head makes a little ball that away it goes with the water. And it allows the water to dissolve the fat. Do you see how powerful water is? Especially if we mix the two together. This is an amazing feat. This is what allows your body to work with so much water and not get dissolved away. Because you're making this barrier. I'm going to use the word barrier. I'm not using the word wall. I will not use the word wall here. Because a cell wall is found in plants and bacteria and fungi. You have no cell walls. Is that clear? I'm looking at you. Is that clear? Is that clear? Okay. Cell walls are different. We'll talk about them tomorrow. Cell membranes are barriers. So if you can think of this brick wall as a wall, think of your membrane like a sheet. Like I put a sheet on a string, right? Your membrane is like a sheet. It can separate two spaces, right? Have you ever made that little forts at home with your bed sheets? So you can be in a room and make a space with a sheet, but it's not a wall. Does that make sense? You imagine it's like a wall. It is a wall. It acts like a wall in many ways, but a wall is much, much stronger, right? Much thicker. It's used to hold things up. It's used to hold things up, right? And it forms a strong separation. Where a membrane, it does separate, but it's not. And it does give you structure. It does. So when you built your little tent fort, it's a tent. You know, it acts like a wall. It does. But nobody's going to confuse this cement wall with a tent, right? There is a difference. So this membrane does create separation. So that's your phospholipids. That's how phospholipids work. That's their strength. That's their strength. If it forms these bilayers, lipid bilayers, they dissolve, they can form micelles. So there's a lot of words there. We're gonna work with that a little bit next week, especially in aquatic areas. Trying to get this stuff out of the way. So that's a phospholipid. Now, who can tell me the difference between a phospholipid and a triglyceride? Go ahead.
A phospholipid has charge and a triglyceride doesn't.
Exactly. The most important difference, a triglyceride first of all, has 3 fats, right? It's just fats, neutral, so there's no charge, so it's nonpolar. Phospholipids have that charge side. Who can explain why saturated fats are solid at room temperature? Because they're not bent from the double bonds, and they can stack one on top of the other. Because that's what they can get real close to one another. The unsaturated ones are bent, and they can't get as close to one another. They can't stack. Relate the structure of phospholipids and their chemical properties and their functional role as a cell membrane. I literally just explained it, so you should be able to answer that. I'm not going to repeat all that. Suggests how the cell membrane structure of an Arctic fish might differ from that of a tropical species. Oh, by the way, cholesterol. So you got, interesting thing about cholesterol is that it's amphipathic as well. In other words, it has 2 sides as well. And it turns out that the cholesterol rings, it has a polar head and and then it has a group of rings. So here's the head. And then the steroid rings hydrocarbon chain, this whole part here tucks into the membrane. So what you get, so imagine this in a membrane. Think about this, because this is really important. You get this, and I'm going to draw cholesterol black. So here's cholesterol with its kind of a little bit of a head here, and then there's these rings. Why are people putting things away? It's 2:09. What time do we leave?
11.
So we got 2 minutes. Can you give me my 2 minutes? So here's the lipid tail. These ring structures that I'm doing a horrible job at drawing, these ring structures are all they're all nonpolar. But the head of cholesterol is polar, so it sticks like that. Sticks like that, actually sticks out, right? So there, it's actually bigger than that, I should draw. But what this does, what cholesterol does in your membranes, you have them in all your cell membranes. All animals have cholesterol. Plants don't have this. Animals have it in their membranes because it helps keep their membranes fluid. It keeps them from freezing. It's like antifreeze. Acts like antifreeze. Should be writing this down because I promise you it'll be on the test. The less people write, the more I want to put those questions on the test. Just remember that. So those moments where you see everybody else writing and you're not writing it down, guarantee you that that's going to be the question I put on the test. So cholesterol acts in the membrane, acts like an antifreeze because you know how butter turns solid at room temperature? But olive oil isn't because of that kink. Imagine if you put a big brick in between the two molecules, stopping them from stacking one another. So the cholesterol literally stops these phospholipids from stacking next to each other, turning into a solid. Because what's freezing? Turning into a solid. So that in winter, a lot of animals collect more cholesterol, and in winter they have more cholesterol in their membranes than they do in the summer. Why is that? Because the more cholesterol you have in your membrane, the more fluid that membrane is. And the membrane has to be fluid. The membrane has to be able to move around. If not, you can't function. You don't want to freeze and break, right? So, cholesterol, what would you expect to see in a fish that lives in the northern oceans?
Like cholesterol?
A lot of cholesterol. You expect to see a lot of cholesterol and any protein that helps them stop the membranes from freezing, from coming together. Explain the structure enables the form structural and functional roles within membranes. Okay, so I just explained it to you. You just saw it. Good luck. See you tomorrow. Tomorrow we will start carbohydrates.